Welcome to another of our daytime lecture series. My name's Irene Crebin. I'm with the Florey's Fundraising and Marketing Department. Before we start this morning, I'd just like to acknowledge the traditional owners of the land that we're on. This event's taking place, the land of the Wurundjeri people, and pay respect to their elders and families. A little bit of housekeeping. Our speaker will speak for probably about 40 minutes, thereabouts, allowing 15 or 20 minutes for questions. If by any chance you really need to go before the questions are over, maybe take the opportunity in that break to quickly duck out so that we don't disturb other people. And of course, could you please turn your phones off, down, or something. Also, on your way out, there are a number of things on the to pick up, including a card, which I didn't bring in with me. You'll see them out there. It's just a little card where we're enabling people to give us some feedback. And what we'd really like from you is any ideas that you have for next year on the lectures. So fill out one of those cards, and you can just leave it there, and we'll take that upstairs. So anything that you've got to say would be welcome. Happy to hear it. Oh, sometimes we're not happy to hear it, but most of the time it's good. And also, Takeka's lovely little book, which she made and printed, featuring her botanical art. It's a great little book for just keeping all your special dates in, anniversaries, birthdays, and so forth. And she's selling that, and all the proceeds come to the Florey. So she's been doing that for years, and this is the last one she's going to do. So it's a rare book, so grab one. A great little gift too. Now, this morning I'm really delighted that again we have a new speaker. I don't think you've heard her before, and she's young, she's female, which we love. And she's also involved with Alzheimer's research. Her name is Dr. Yen Ying Lim, and she's an Alzheimer's researcher. She's got a fellowship here at the Florey. We're delighted to have her. And she heads up a project called the Healthy Brain Project. And if you're a football follower, and a couple of weeks ago you were watching, you might have seen a lot of information about the Healthy Brain Project in conjunction with a Collingwood football game, because Eddie Maguire and the Collingwood club are supporting the Florey, and they gave us a chance to promote that. Yen's primary interest is in understanding how genetic and lifestyle factors independently or interactively accelerate or protect against brain diseases, and particularly dementia. So she's supported by the National Institute for Dementia Research and the Alzheimer's Association of the USA. Is that correct? Right. So we love this kind of work, and we love a young person getting stuck into it, because I'll speak for myself, I need it, and maybe we all do. So I'm not going to say any more, but introduce and welcome Yen.
Good morning, everyone. Thank you so much for having me. It's the first time I've done something like this. I will try not to speak for too long so that you guys have an opportunity to ask us some questions at the end of it. I thought, I just arrived back from the States yesterday. I was away at a genetics workshop, and I was trying to think about what to put together for this talk, and I thought, let's focus in a little bit about just give you guys a brief history about Alzheimer's disease. I'd like to talk a little bit about the genetics of the disease. I get a lot of questions about, my mom or my grandmother has Alzheimer's disease. Does it mean that I'm going to get it? What is my risk? So I thought I'd give you guys a little overview of that, and then I'd like to sort of end up with a summary of where the treatments and therapies are at the moment, and what we know about the disease, and where to go forward from here. So a lot of you, this does not work. Sorry about that. So dementia is really an overarching umbrella term that we use to describe a loss of general cognitive function or memory and thinking function over time. There are different sorts of dementia. Alzheimer's disease is the most common form of dementia, where about 60 to 80% of cases that are diagnosed as dementia are actually Alzheimer's disease. But there are obviously other forms of dementia as well, like vascular dementia or Parkinson's disease, dementia of the Lewy bodies, or frontal temporal dementia. These are all the different forms of diseases that can go wrong in an aging brain. Alzheimer's disease is the most common form of it, as I said before, and that is really where I have focused my research on. There are a couple of really alarming statistics. Don't really know why this is not working. Okay, there are a couple of really alarming statistics about dementia. It's the second leading cause of death in Australia, and there is no cure. So it's just right behind cancer on that. But you know, we really don't have anything that can stop it or stem its progression. As you can see, over 300,000 Australians are living with dementia, and that number is projected to triple by 2050, and that's really because of our aging population. So we're living longer. Medical research is doing such a great job at keeping us alive for a lot longer, and that in turn increases our risk for developing dementia in the future. The worldwide cost of dementia is absolutely staggering. It's about $815 billion US dollars worldwide. And the rate of Alzheimer's disease also doubles every 5 years after the age of 65, so reaching about 30 to 50% once people reach the age of 85. And all that, you know, all that sort of sounds really scary. And like I said before, that's really because of our ability to now live for a lot longer, and age is really the biggest risk factor for dementia. So this is just you know, a graphic to show our worldwide prevalence of dementia, the predicted % increase in Alzheimer's disease by 2050. And so in Australia down there, there's 365% of you know, increase by 2050. Despite that, spending on dementia research is, you know, not great. I like to put this graphic up because I think it kind of illustrates how we spend more each year on popcorn than we do on dementia research, which is quite phenomenal. I mean, you know, part of that is because Hoyts charges us, you know, absolutely crazy prices for popcorn, but still, I think it puts it all into perspective a little bit about how much we actually spend on dementia research. The first case of Alzheimer's disease was described by Dr. Alois Alzheimer, so the disease was in turn named after him. This was the case of Auguste D. in Germany, I think it was. And he described this woman who was about the age of 53 at that point, with profound memory loss, unfounded suspicions about her family, so a lot of paranoia, and other worsening psychological changes. When he studied her brain at autopsy, there was a dramatic shrinkage and abnormal deposits in and around the nerve cells. And even to this day, that is how we can only definitively diagnose dementia, Alzheimer's disease, and that is at autopsy. We can talk about, you know, probable Alzheimer's disease, possible Alzheimer's disease, but it's really only at autopsy that we can definitively say that this person died from Alzheimer's disease. So clinically, the symptoms of Alzheimer's disease obviously are characterized by memory loss, and memory loss is the key thing in Alzheimer's disease, and it's a key component of all the different cognitive functions that we study. There are challenges in planning or solving problems, and as the disease progresses, there will be, you know, increasing confusion about time and place and inability to then dress themselves and conduct basic activities of daily living. Alzheimer's disease is characterized by two main types of pathologies in the brain, amyloid plaques, so there are these sticky plaques in the brain, and also tau tangles. So there are these two things that we look for when we diagnose somebody with Alzheimer's disease. On my right is a normal brain. And beside it is a comparison of an Alzheimer's brain. So in the normal brain, you can see that the brain tissue is quite healthy. It's quite packed in there. Whereas with the Alzheimer's brain, there's quite a bit of atrophy, a degradation of brain cells around it, such that there are big spaces in the brain. Let's see. This here? Nope, doesn't work. Sorry. So this here is a neuron, and these are normal nerve cells. And inside the cell, what we find are the tangles, so the neurofibrillary tangles, and outside the cells are what we try to look for, the amyloid plaques, so these sticky clumps of protein. And those two are the hallmarks of Alzheimer's disease. Here is another image of a neuron, so a nerve cell that has been treated with an amyloid plaque. And so this white sticky substance here is what it looks like, and this has been, I think this won the prize in 2012 for science photography, and it's one of my favorite images because it's so pretty. Again, this is what an Alzheimer's brain looks like compared to a normal brain. So you look at all the big spaces in between the folds, and that's what happens when as the disease sort of progresses. To try and understand the cascade of things that happen across Alzheimer's disease, a lot of people have tried to model what we think happens. It's understood to have a very long preclinical period with gradual changes in neuropathology and cognition. So on the vertical axis is the biomarker magnitude, so the magnitude of different changes like cognition, like brain imaging, tau, different things like that we can measure in a person. And on the horizontal axis is the clinical disease stage, so you go from normal all the way to disease. The first thing that happens is pathologically, so as I said, Alzheimer's disease is understood to be characterized first by the accumulation of amyloid plaques in the brain, which can be detected both by either a brain scan, so a PET scan, or by taking a spinal tap and taking some fluid from your spine. That will determine the level of amyloid in your brain. And so, as you go from normal to abnormal, normal to Alzheimer's disease, that increases over time. This is then followed by changes in tau-mediated neuronal injury and dysfunction, so that the second protein starts to develop, starts to accumulate over time. And then followed by neurodegeneration, which can also be detected by which is basically loss of neuronal structures, particularly in the hippocampus, and that can be detected by MRI. So it's a different type of brain scan that we can do. And the thing to note is that these can only be detected closer to the disease stage. As you approach the point at which a person will come and present to a clinic, so the point at which a person starts to complain about their memory, starts to notice changes in their memory and thinking function, this is when we can start to detect changes, very subtle but significant changes in their ability to recall information, and also do a couple of other tests, like ability to plan things ahead of time, their language abilities start to deteriorate, and also their attention as well. Finally, as a person enters the disease stage, enters a clinical presentation of Alzheimer's disease itself, their ability to manage finances, to cook by themselves, to plan, to clean, and to look after their own personal hygiene starts to deteriorate. And that's where the full-blown sort of syndrome of the disease starts to present itself. Age is the largest risk factor for Alzheimer's disease. This is a figure showing the prevalence of the disease at each age group. And so as you can see, from 65 to 90 plus, the prevalence of Alzheimer's disease increases So the older you are, the higher chances you have of developing this disease. And that is irrespective of any kind of race or nationality. This again shows you that the prevalence of Alzheimer's disease increases with age, and the prevalence of plaques, so the amyloid plaques, in healthy people also increases with age. And so the biggest risk factor for developing these amyloid plaques is really age as well. Why are there plaques in healthy adults? That has been something that has plagued us for quite a while now. And it really has given birth to this idea that some people can have a head full of plaques, a head full of Alzheimer's pathology in their brain, and yet still be clinically healthy. And so that really, really came up with, so scientists really then came up with this idea of cognitive reserve or resilience, which is then an ability somehow, due to some combination of genetic or lifestyle factors, to be able to protect the brain against the disease despite having a head full of pathology in their brains. This is post-mortem data, so post-mortem autopsy information looking at the prevalence of amyloid again in the brain, and again showing that there is pathology without clinical expression. So there's pathology in the brains of some people, but despite that they can remain clinically healthy, and that makes them a really, really interesting group to study, because if we can understand what it is about these people that make them healthy, despite having so much Alzheimer's gunk in their brain, that might help lead us to some kind of therapy or treatment or intervention that might be efficacious. So there's a global effort to understand Alzheimer's disease. Everyone is trying to understand this. The European Prevention for Alzheimer's Disease Consortium, the Alzheimer's Research UK, America there are a lot of different studies in America that have been trying to understand Alzheimer's disease and treat it. And in Australia, we have our very own Australian Imaging Biomarkers and Lifestyle Study, ABLE, which I think some of you may be a part of, and the recently launched Healthy Brain Project, which Dr. Rachel Buckley and I have started to try and understand the very, very earliest stages of this disease. Our ability to track the course of the disease has been improved markedly by neuroimaging methods. So these are different PET scans that we've done on people with Alzheimer's disease, people with mild cognitive impairment, and people who are healthy. So the warmer the color, the higher the level of amyloid plaques. SUVR is really just a metric that we use to measure the amount of amyloid there is in the brain. And so if you look at the top picture, the top image of a person with Alzheimer's disease, you can see that it's very, very warm with lots of signal and amyloid plaques. Coming down on a person with mild cognitive impairment, so someone who is in the prodromal stage of the disease, so someone who doesn't quite have the full-blown clinical syndrome yet, but it's on their way there. You can see that it's slightly less warm, but you can definitely tell that the amyloid plaques are really lighting up. And then in healthy older adults, which are the two bottom scans, the amyloid positive, what we would consider someone who is amyloid positive, an amyloid positive healthy older adult, you can see that there's still some signal. It's still quite warm. The colors are still quite warm there. So it would suggest that these people may be on the preclinical stage. So really, they're still clinically normal. They're still healthy. But they're on their way to developing the disease. And you contrast that with the bottom scan, which is someone who we would consider amyloid negative, so someone who has no, very, very low levels of these amyloid plaques in their brain. We would consider that bottom scan to be someone who is pretty normal, and everything's all good with them. So this led to the recognition of what we call preclinical Alzheimer's disease. And so, if the bottom, so with increasing years, The dotted line shows the trajectory of cognitive function as somebody progresses from clinically normal all the way to the dementia stages. And so this very early stage here, which is early symptomatic, amyloid positive, cognitively normal older adults, if we follow them over time, we can detect very subtle but very significant memory changes, suggesting that they're very slowly they're very slowly on their way to developing dementia in the future. It's not 100%. I need to really stress that to you guys. It's not 100%. So just because you're amyloid positive does not mean that you will develop the disease, but it increases your risk substantially. So we've talked a lot about sort of risk factors, genetic risk factors, things like that. The thing with trying to understand disease and genes is that a lot of the times, rare genetic mutations have the most impact on genes. So, for example, these genes here, APP, PS1, and PS2, they cause Alzheimer's disease. If you have a mutation in one of those genes, you will get the disease, but they're very, very rare. So it's 1 to 2% of the entire Alzheimer's population will have these mutations. On the other hand, you have very common genes that might not necessarily play much of an impact on different things, but together they may interact with each other. And so a lot of my research is focused on understanding these different genetic risk factors and how they play into the scheme of things. But before I talk more about genetic risk factors of Alzheimer's disease, I'd like to back up to Gregor Mendel and his Ps. So this is just a really quick, very, very quick overview of Mendelian genetics. Gregor Mendel is an Augustinian monk and a part-time scientist. And what he would do was notice that sometimes the peas in his gardens would have yellow peas, and sometimes there would be green peas. And he sought to sort of understand that process a little bit more. So I thought I'd give you a very, very quick tutorial on how genes kind of work. Some of you probably know this already and can probably explain it a lot better than I can, but I will try. So I want you to focus on the right-hand side of the screen. So just here, if you have a purebred yellow pea and a purebred green pea, and you cross them, so each gene will have what we call two alleles, so two variants. So a purebred yellow pea will have two capital Y variants, and the purebred green pea will have two lowercase y variants. And if you cross them, you get a capital Y variant and a lowercase y variant. But the thing with the capital Y variant is that it is a dominant gene. And so with the dominant gene, that trait will be expressed. Right. So when you cross the yellow pea with the green pea, there are no green peas because the yellow variant is the dominant variant, and that is expressed as a yellow pea. If you then cross the two second-generation yellow peas that have a capital Y and a lowercase y, what you get is, this is what we call a Punnett square, so it's trying to work out the probability of the expression of different types of variants or alleles. What we get is one purebred yellow pea, one mixed bred yellow pea, another mixed bred yellow pea, so two mixed bred yellow peas, and one purebred green pea. And that's because of the difference. So you inherit one from the mother pea, one from the father pea, one from the father pea, one from the mother pea. And that's how you get 3 yellow peas and one green pea. And through that way, he was able to work out this idea of dominance and also inheritance. That really kind of formed, that idea was very, very controversial in the 1800s, but he was really the father of sort of modern genetics and how we understand inheritance these days. So coming back to genetic risk factors, these genes here, APP, PS1, and PS2, these 3 genes that cause Alzheimer's disease, they are dominantly inherited genes. And so we consider these patients to have autosomal dominant Alzheimer's disease. It's also known as dominantly inherited Alzheimer's disease, and so if you carry one of these mutations, you will get the disease. Sometimes it's also known as familial Alzheimer's disease. As I said before, it makes up very, very small cases, small numbers of Alzheimer's cases, so only 1 to 2% of Alzheimer's cases have dominantly inherited Alzheimer's disease. It's also inaccurately called early onset Alzheimer's disease. I want to make this really, really clear because I know a lot of people have come up to me and said, oh, my mom or dad developed Alzheimer's disease in their 50s or 60s. I think they have early onset Alzheimer's disease. I don't like to use the term early onset because it's really non-specific. With autosomal dominant Alzheimer's disease, the average age of disease onset is about 40. So I remember sitting in a clinic with patients with autosomal dominant Alzheimer's disease, and it was this mother who had just had a baby a year ago, but she couldn't remember who her baby was anymore, and it's absolutely heartbreaking. But this form or this variant of Alzheimer's disease can be detected really only by genetic test. And if you carry one of these genes, you have this disease. You have 99.9% chance of developing this disease if you test positive for this gene. And I say 99.9% because so far I think there has only been one case, one person who has this mutation but has not gone on to develop the disease. And he feels like he's won the lottery every single year, and it's pretty amazing. So we have him under study. We're trying to understand what exactly it is about him that makes him so special. There's only one person. The most devastating thing, though, is that there is a 50% chance of passing this on to your children by that whole dominantly inherited pathway. So this is what a family tree of somebody with autosomal dominant Alzheimer's disease looks like. So these are brothers and sisters. Black circles are the ones who have the disease. Brothers and sisters who have the disease, they get married, they have children, and 50% of their children have the disease, who then pass on to 50% of their children. So you will know that you have this disease because about half of your family will be completely ravaged by it. And so if you know somebody, or you yourself know, you think you might belong to one of these families, or you know somebody who might belong to one of these families, please let us know. We can put you in touch with different research groups that are currently studying these groups. So the Dominantly Inherited Alzheimer's Network is one of the biggest groups that's looking and trying to understand this really rare but really aggressive form of the disease. And they have a clinical trial going on at the moment as well. And so the first case of Alzheimer's disease, Auguste, was, we now think that she has a form of this disease, of autosomal dominant Alzheimer's disease. It's really important for us to study this very rare genetic form of the disease because it gives us clues as to what happens in what we call sporadic Alzheimer's disease, so the large majority of Alzheimer's disease cases. Because these guys are so young, they start to develop the disease at around 35 to 40 years old. They are so young, but it also makes the disease so pure. There are no other competing diseases of aging, other aging factors that impact on their disease because they're so young, and it makes them really, really important for us to understand. So in terms of the other 98% of the Alzheimer's population, what about them? What about their genetic risk factors? The only conclusive genetic risk factor that we've been able to identify is the apolipoprotein E, or ApoE, E4 allele or variant, and that increases the risk of developing Alzheimer's disease. So sporadic Alzheimer's disease, sometimes called late onset. Again, I don't like to use the term late onset because you can have sporadic Alzheimer's disease but you can have early onset. Age is the greatest risk factor for this. And the greatest genetic risk factor is the ApoE4 variant. So it increases your risk of developing the disease by about 3 to 4 times. So ApoE can take 3 variations. There's E2, E3, and E4. And then that makes 6 different possible combinations. So every person has 2 variants, so you can be E2, 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 E3, E2, E4. The one that has the highest risk is E4, E4. So if you have 2 copies of the E4 allele, your risk of developing the disease is about 10 times. But again, it's very, very rare to have 2 copies. It is, however, a lot more common to have 1 copy. 1 copy will increase your risk by about 3 to 4 times, and it's present in about 15 to 20% of the population. So it's not a rare thing. There is some family history component to sporadic Alzheimer's disease. So if you have a family history, you have a higher chance of developing the disease, but that's through the different alleles. But it's not 100%. It's definitely not like autosomal dominant Alzheimer's disease at all. Interestingly, E4, ApoE4, is the ancestral allele, right? So a lot of people are like, well, if our bodies and evolution is so efficient, why is E4, if it causes us so much harm, why is it still around? Well, E4 is the ancestral allele, and over time, E3 has become the most common form that people carry. So E3 here is what we call the neutral form, and that about 79% of the population will have that. Right, so that over time that has become the most common form. But traditionally, ancestrally, E4 is the allele that we have, and we think that it plays an important role in lipid metabolism. And so, in times of famine and things, it becomes very helpful for us to have this allele. But obviously, as we as society has evolved and we move away from hunting and gathering and things like that, and famine becomes less and less of a problem, it becomes quite maladaptive after a while. So the effect of E4, what does E4 do, and why does it increase risk of Alzheimer's disease? The main effect that E4 does is on amyloid accumulation. So we think what happens is that amyloid disrupts clearance of amyloid. E4 disrupts clearance of amyloid. Amyloid plaques build up in our brain over time. Everyone has some level of amyloid plaques in our brain. It's just kind of like plaques in our teeth. Everyone has them, but you brush your teeth and you get rid of them. Just like with the E4, there is a clearing mechanism that clears it from your brain. If you carry the E4 allele, it's kind of like being born with a faulty toothbrush. You can't clear the amyloid plaques from your brain as efficiently as carrying the other two alleles. And so what we've done is start to plot the levels of the rate of amyloid accumulation in different groups of people. And what we find is that if you're an E4 carrier, you accumulate amyloid about 17 years faster than if you carry the other two alleles. And so that's current understanding of how E4 plays into the mechanism of Alzheimer's disease. There are a lot of other functions that it does as well, but amyloid, its role in clearance of amyloid seems to be a key one. In terms of treating Alzheimer's disease, the aim is really to stop amyloid accumulation. So the current prevailing theory is that amyloid precedes everything, and it results in a cascade of events over time. So we want to try and stop amyloid accumulation before it even starts. So there are different types of therapies that are being developed. Some enter the synapse and interfere with cell-to-cell transmission of amyloid. Some bind to amyloid to promote removal. Some bind to amyloid oligomers and promote clearing. So there are all these different mechanisms that people are trying to look into to try and get rid of amyloid. However, it's been an absolute graveyard of Alzheimer's therapies. Every single one of them have failed. And it's quite depressing to look at this. Most recently, there was the BAPINEUZUMAB failure. So it failed to moderate or fail to result in any benefit to patients, and these are huge numbers, huge clinical trials. and they failed to find any benefit. Strike 2 was SOLANEZUMAB, so Eli Lilly's drug, didn't find any effect on cognitive endpoints. It did manage, they say that it did manage to shift sort of amyloid levels, but the thing with the FDA, so the governing body of drugs in America, what they say is that if you fail to show any benefit on a person's memory function or their ability to conduct daily activities of living, then your drug is no good to us. You can show me as many pretty pictures on imaging as you want, you can shift as much amyloid as you want, but if it doesn't result in a benefit to the patient, then it's not an efficacious drug. And so that's sort of the attitude towards all these things. So that was Expedition 1, and that failed, and then recently Expedition 2 failed as well. So there was no benefit to participants. There is some hope. So Biogen now has a drug called ADUCANUMAB. They presented their phase 2 findings a couple of years ago. And what they were able to show was a dosage effect. So the higher the dose, the lower the amyloid levels, but also they were able to lower the amyloid levels with higher doses of drug, but also they were able to shift the endpoint, so shift the memory and thinking function of these participants. So the blue line here shows the placebo group. Sorry, the placebo group is shown in the orange line, and the blue line is shown in the treatment group. And so they were able to show that. However, there is always a however. With ADUCANUMAB, it is a very aggressive drug, and the concern here is what we call ARIA, or they get a lot of hemorrhages in these people who take this drug, and that's because amyloid tends to clump around vessel walls, and so when you remove them, there is a chance that people might stroke out. So it's really quite a catch-22 that we're sort of faced with at the moment. Another reason why we think that these drugs are failing is because we're treating it too late. So when somebody has Alzheimer's disease, when somebody's brain is already half gone, and from all the amyloid plaques and tau tangles in there, we are not able to bring back brain cells. There is no way that we can bring back brain cells anymore. And so there is now a shift in attitude towards what we call secondary prevention, even primary prevention. So secondary prevention is identifying people who are amyloid positive, so who have abnormal levels of amyloid in their brain, but they're still clinically healthy, so they're not quite so advanced in the disease yet, and treating them then. Early detection, early treatment, we're hoping that will work. There is now we're going to try and push that even further into what we call primary prevention. So identifying people who are genetically at risk, because genes come first, they always come, you're born with them, so they always come first. Identifying people who are genetically at risk and seeing if we can stop amyloid accumulation before it even starts. So in terms of secondary prevention, there's the A4 study, which is a large global study run out of Harvard. It's called the Anti-Amyloid for Asymptomatic Alzheimer's Disease. We have one of the largest sites here in Melbourne, and so if you're interested in that, check out their website or give them a call. And they're currently recruiting participants to be a part of this trial. So you have to be clinically healthy, so cognitively normal, but you undertake a brain scan to identify whether you have high levels of amyloid or not. And then if you do, then you get enrolled into the trial. And we're hoping that is early enough to sort of catch it. Moving into primary prevention, though, which is sort of where a lot of my work is starting to head. This is a paper that we just published in the journal Neurology. And if you look at these are very young people, so they're age 20 to 70 years old, very, very, very young, before any kind of abnormal levels of amyloid are present. And these are people who carried ApoE2 or E3, so the neutral genes, and these are people who carry ApoE4, so they carry the risk gene for Alzheimer's disease. And if we follow them over time, if we measure the amyloid levels over time, we can see that they're starting to accumulate amyloid, even though it doesn't reach levels of abnormality just yet. The disease process is happening. We can detect it very, very early on in middle-aged adults, even though they do not reach any threshold for abnormality. And this really is the inspiration behind setting up the Healthy Brain Project. So the Healthy Brain Project aims to recruit people aged 40 to 65 years old. It's an online study because we want to make it as accessible to as many people as possible. I am really proud to say that about 40% of our participants live outside a major metropolitan city, which is great. I think that's really important for us to be able to reach out to people who don't live in Melbourne, Sydney, or anywhere like that. They enroll into the study, so you sign up. You do this all online. People who are aged 40 to 65 are very, very, very busy. They do not have time to come and spend 3 or 4 hours in the clinic with me doing a bunch of testing. So they can do this all online at their own time. And when they finish, what we do is we send out a saliva sampling kit in the mail to them, and they just spit it into a tube, and we can get some information on their genes. So when you sign up to the study, this is what it looks like. This is your profile when you get in, and on the left-hand tab there are some surveys and different types of thinking tests that we put our participants through. There's also a forum that we want to encourage some discussion about how their experiences with the disease, what they'd like to know more of. They can ask us questions, different things like that. Primary prevention is so important because what we found is that a large proportion of risk factors for Alzheimer's disease is actually quite modifiable. So diabetes, midlife hypertension, physical inactivity, depression, smoking, and low educational attainment. These are all sort of risk factors for the disease. The risk is not very big, so if you look at the relative risk, it's about 1.6 times, 1.8 times, so it's not huge, but there are things that we can do. Genes we might not be able to change, but lifestyle we most certainly can. And so promoting and understanding how these lifestyle factors, especially in midlife, sort of affect the development of the disease is really a key aim of our study. So just before I kind of wrap up and end this, I get a lot of questions about brain training. What about brain training? What about, I do Sudoku, or what about lumosity, things like that. I don't know how aware you guys are, but lumosity just a couple of years ago was sued and had to pay out a huge amount of money because they were claiming things that weren't true. And that is the issue really about a lot of these brain training games. They claim to say that they claim things like, it decreases your brain age from 78 to 26 in 3 days. Like, that's ridiculous. Biology does not work like that. Our bodies do not work like that. It takes a long time. You don't just decrease your age by decades in 3 days. And so the users may get this illusion of very huge gains when in fact it's not true. What they're looking at is really what we call a practice effect. So if I give you a test and I give you the same test over and over and over again, you are going to get better at that test just because you've practiced it. That does not mean, though, that you are able to translate that gain into something else, into remembering your shopping list when you go grocery shopping, or remembering where your keys are, or anything like that. And so a lot of the brain training industry, it's a multi-billion-dollar industry. They have really capitalized on the fear that a lot of people have about their memories and turned it into a lot of money. So, so just be really, really careful if you want to subscribe to this, something like that. But what can you do instead, right? Like, all the good things that my grandmother tells me to do. So eat a diet low in fat and cholesterol, rich in dark-skinned fruit and vegetables. Stay mentally active, stay socially active, stay physically active. Get enough sleep. Sleep is a really important one. You don't have to go overboard and sleep 9, 10, 12 hours or anything like that, but just get enough sleep such that you wake up feeling refreshed. I think in our ever frantic world, there is almost this attitude that, like the less sleep I get, it's almost like a bragging thing, right? Like, I only got 4 hours of sleep last night. That is not a good thing. Try and get enough sleep. And if you really, really, really want to, try and learn something new instead of subscribing to a brain training app or anything like that. Try and learn a new language, learn a new skill, pick up the piano again, do things like that, play bridge. Those things will serve you a lot better than downloading an app and paying hundreds of dollars a year on that. So thank you very much for your time. Thank you for listening to me talk about all these various aspects of Alzheimer's disease. You can help us by signing up to the Healthy Brain Project if you'd like to. And we've got some information out there for you as well. Thank you.